Okay, let's go ahead and get started. Whatever is the most confusing things, we'll just walk through it again. So same thing, we always go around the table. And if you have a question, we'll ask the question, and if not, you can skip.
Okay. I guess. So I guess I'll just start with like the other business experiment. I think I'm a little bit confused about the experimental setup. So
Let's talk about
how do they ensure that the 796 motor and 722 motor are on the other side, on either side of the helix, and like vice versa?
So this is gonna be primary design. Remember we talked about hybridization and that we actually make a, what you call it? We make, what's the word?
The probe?
We make a probe that's specific to a spot on the DNA.
Yeah.
So we think of what the actual tube looks like, and then so this here can be a little bit misleading. So I'm gonna draw it how the actual experiment happens.
Okay.
They will have two different test tubes. They have one test tube here. I think this marker's darker now. Does the blue show up? Yeah, we're gonna have a test tube here. We're gonna have a test tube here. In this test tube, we're gonna add a helicase, so this is gonna be our helicase. Add a helicase here. We're actually gonna put in our long strand. We're gonna put in our long strand. And because we have open ends, the helicase can get onto the DNA. One of these is gonna have the 722 mer. The other one's gonna have the 796.
Yeah. Oh, gotcha. Okay, that's how.
And that's how they make sure only one is there at a time.
Oh, okay.
And then they do the reaction, you run the gel. And of course, we expect this on lane one, that if it's up here, then nothing got displaced.
Right.
And then if it's over here, if it got displaced, which is what we saw in the example, then this helicase has to go from this direction, this direction. Because we designed these ourselves, we know the 5' to 3' end.
Right, okay, no, that makes sense to me. I thought they were on at the same time, so I was really confused about how they could do that.
Yeah, no, they can't be that accurate.
Okay.
So they're separated into two different tubes.
Okay, my bad, thank you.
Okay, so is this clear? Yes.
So like, where does the fragment at the bottom come from, the one that gets displaced?
So the fragment at the bottom comes from the fact that it is displaced. So remember when we do agarose gels, it's kind of like size exclusion. Whatever's at the top is big, whatever is at the bottom is small. So since it's little and it gets kicked off, it's going to show up at the bottom of the gel.
Okay, so what's being labeled is like a smaller.
A smaller piece, yes. Any other questions on this experiment? You will have a question on it, so note it. Yes,
And let's flip to the other side of the bottom, then we go 3 to 5 times, that's right.
so if it looked like this is a question. So I have a eraser. Ha. Would this now be a 3 to 5' helicase?
Yeah.
Yes, exactly, it would be. If you saw this, what's this? What do you think happens here?
Forgot that.
Yeah, you didn't add any helicase, or a helicase broke, or there's a mutation that doesn't work, right? Something, the helicase doesn't work. What if you saw this? It can go both directions. So kind of really clear, right? Once you get the concept.
Yeah.
Okay, what if you saw, we'll do one more. What if you saw this?
Forgot to put it.
You forgot to put it. Exactly, good. So understanding like how this is set up will make the experiment really, really easy.
You forgot to put in, oh, they're not getting.
You forgot to put in the long visual DNA strand.
That's super helpful, because I thought I was like, how do they get it to?
So like, how do you get it to pick and like, yeah, okay. So that feels clear now, yes?
So it contains like a long transfer and then just a little piece that's labeled.
Yes, a little piece that's labeled at each end, one at a time.
Yeah, thank you.
Okay, any other questions on this concept? Yes,
So is the top strand is just the long strand, and so
so the top strand is the long strand and the
it's
label.
not marked, it's not labeled.
It's not, it is, the only way we see it is because of the label. This is like the radio label. So this is some of it that's still associated at the top.
But then when the case goes to 500 direction, it separates it, and that will top right.
It displaces and falls off. So let's also think about, remember how we talked about things being 100% efficient or not efficient? In almost any experiment, you assume not 100% efficiency. But say in this end, we forgot to add this, but also technically, if for some reason helicase could take all of it off, 100% efficient and you can't see it. So that's another scenario where this gel could look like this. but that's not normal. You always have some left behind and both will be related. And so what I'm going for is a deep understanding of how these experiments work, how you interpret the data. And then when you get to your upper divs, they'll give you like, here's a figure, I need you to understand this, and then like write out what you mean, draw a schematic, and there's no like drop down menu to pick what you want. So you need to understand, and they're not gonna be as nice. And then it's gonna be rough if you're not critically thinking. And if you do this, like you go to your next class, you go, oh, wow, this is straightforward. Okay, I got this. And that's what I want for you. Okay, any other questions on this topic? Yes.
You said that if the displacement was for the 796 mark, it would be still 5 prime and 3 prime. Why is that?
It's gonna be 3 prime to 5.
Oh.
Yeah.
um,
So just remember direction, whichever one's attached to, and then decide that it went.
If you go in both sides, if they both join like 2.
Mhm. Is this one clear? Yay, okay. We are where we're at now here.
I have a question where RNA is removed on nuclear device and then to the DNA, so if you're on the new strand and then bonded directly to your RNA fiber, is there anything externally in it?
You mean like, here is, so say this is parent. And then say this is the daughter. Here, 5 prime, 3 prime, 3 prime, so 5 prime. You're saying this is like the RNA, right? You're asking if RNA can just bind here, remove the, yes, it can. Okay, next question? Nope. This one? Next one. Yeah, we know stuff, okay. Sorry.
I have one on the quiz, it was regarding the question I read, so it's like the function of DNA ligase is to, and there's a select all apply.
Mhm,
So the first one is catalyze the formation of covalent bonds between the adjacent nucleotides, that's correct.
mhm.
Join off-consecutive fragments with other pieces of DNA, that's correct. And then catalyze, catalyze the formation of hydrogen bonds between adjacent nucleotides, that's not correct. But then the last one, add the missing nucleotide between 2 off-consecutive fragments. Oh, I guess ligase doesn't add a nucleotide, it's active phosphate. Okay, I have to
Yes.
do that.
So
Okay.
remember, that's good, but that's the, like, reading stuff a lot actually helps. So I've only been noticed when, some of you might have just randomly got like another 1.5 or something on the exam. Some of you saw that? So I was making the video to go across the answers, when I was reading the answer out loud, I'm like, oh, this is correct. So I had to go fix it really quickly in the key. And then, so I'll drop the exam question. I'll also reiterate which one got like regraded. So everyone who got it right and it was marked wrong, you got your point back. If you had originally gotten it wrong, we didn't deduct more points. Okay, so yeah, remember DNA ligase doesn't add any new nucleotides, it's just making that phosphodiester bond and using intermediates in order to make that happen.
Awesome.
Okay, next question. Yeah, I hope today it should have felt a little bit more chill. Because I know like going up towards exam, it's always a lot of stress. Okay, yes.
Well, I think it's a mechanism for language, you know,
Okay, so let's say this is ligase. And in its active site, it has a peptide group. The part that you need to know is important, that it's just going to interact with ATP. Remember, ATP has a triphosphate bond. By interacting with it, it's going to kick off, so almost the same mechanism in DNA extension where you have nucleophilic attack and you kick off a pyrophosphate. And then you can ignore that, but of course you're going to have pyrophosphatase, give you 2 inorganic phosphates and tetrodotoxin. And then so what you get from this interaction is now you have a ligase that is connected to an AMP. So that's the first step, is use ATP, kick off 2 phosphates and get a ligase AMP. So that's step clear. Okay, now we have ligase AMP. By having a ligase AMP, now we're able to basically cause an interaction on the DNA strand. So let's say this is the NIC. I'm going to say this is 5 prime. This is 5 prime. At the 5 prime side, there should be 1 phosphate. At the 3 prime side, there is your OH. And we want to be able to make this phosphate energetically favorable to nucleophilic attack. So to do that, the ligase actually interacts with the phosphate here. And then you're adding the AMP to the phosphate. And then kicking off the ligase. And I've lost the ligase, and we've created an energetically favorable. Like adduct or, there's a word for it. Yeah, who has their notes open? There's a word for this complex.
Identity mix complex.
Yes, you have your adenylated Nick complex. So obviously you can tell you don't need to know that word because I can't even remember it. So adenylated Nick complex. And what's important about this complex is that it's able to pull enough electrons from this phosphate group to allow the hydroxyl to do nucleophilic attack. So when nucleophilic attack happens, we can now form a phosphodiester bond and this AMP gets kicked off. Or ADP gets kicked off. AMP, it's one. So this makes sense. So it's almost like it's trying to create a mimic of the DNA mechanism of ligation. All right, cool.
So it kind of hijacks the AMP from the ACP. I mean, it takes off the two phosphates.
Phosphates and then hijacks the AMP to make that that phosphate energetically favorable to attack. Exactly.
Thank
Yes.
Yeah.
Yeah. And so you know what's wild about this whole thing? It confused the crap out of me forever. And it's like I don't understand how it keeps getting shorter. And the thing is, in the book, it doesn't actually show the shortening, it shows the elongation. So let's go through it talking about replication. So let's say we have, this is our 3' overhang. So we send it 5' to 3'. This is our 3' to 5'. And so say this is our starting point for DNA replication. That's not, that didn't match. 5', 3'. Say it's our starting point for DNA replication. So if we're going to try to replicate it, we will split it apart, of course, and then we're going to have to here, this is 3', 5'. This is 3', 5'. And then we're going to have to replicate in the 5 to 3' direction. So we're going to add a primer here and extend this way. And then opposite. This is why I draw things. Adding a primer here, which is fine, it's great, and extend this way. And then that's not going to be a problem. Then on this end, we're actually adding a 5, 3'. Okay, cool. We're adding a primer here.
You don't need a slack. It doesn't matter.
Yeah, we should draw on the inside. That'll make it easier to follow. Okay. And then we're gonna have separate look at leading strand, lagging strand. If we're gonna go replication fork follows this way. 5', 3'. Okay, so this is the one that's occurring. This is the, what you call it? Y'all help me out. This is a lagging strand. So it's gonna be leading strand, label. Leading is going to be lagging. And then the replication fork has to move this way, but it's trying to replicate in that direction. So this is going to have our Okazaki fragments. And kind of go here, and go here, and kind of go here. And then depending on where we're at, we'll start replicating. And then especially over here, depending on how big that primase is, or how big that primer is, the primer ends up getting deleted. You have the same amount of overhang. Then on this end, on the other side, same thing is happening and the primer gets deleted. And then every time that we replicate, we use a little bit more based off of like where that primer was first binding. Does that make any sense?
Yeah.
So
Because there's no available.
exactly.
Doesn't have a lagging trend as well on the last.
Because only one on that. Yeah, on the left.
Yeah.
Only one.
Yeah.
But you're gonna find three.
Yeah.
300.
So this is the very end, and then this whole thing's gonna happen at the very end on the other side. And then depending on where like the primer simply starts, so sometimes the primer can sit at the start at the very end. Sometimes you start replicating and then the primer is like right here. It just all depends on like where that primer gets set. Whatever's behind it is not going to get replicated. And then so you can start losing information every single time you divide. Yeah, so when I say it's just like a concept that was that I struggled with and why I said like take that figure, go through it over and over again until it makes sense to you, work on it until it makes sense to you. It's not, it was never straightforward to me. I actually, like when I took this class, I worked my butt off. I got a B. I didn't take it here. I worked my butt off and I was really, really proud of my B. Because for me, it was difficult. And I think it's part of the reason why I'm trying to break it down as much as I can, so I feel like the struggle the same as I did, because I feel like stuff was just confusing me. But now, like, the more I do it in lab now, it makes more sense, and if you do it in your labs over afterwards, it'll also make a lot of sense. So hopefully this is clear. If it's not clear, we can come back to it. Yes,
The one thing I'm confused by is how the telomeres prevent the loss of.
and so technically, that shortening of the DNA still happens. But the telomeres don't have genetic information coded, they're just like random repeats. So if you lose the random repeats, you're not gonna lose the important stuff. Think of it as like a buffer or something.
I see, okay, so that you kind of overhang is part of the buffers.
Yeah, so usually your overhangs will all be like telomere repeats. Mhm,
Including the overhang.
yes,
Okay, oh,
Yeah.
I gotcha.
okay. Any other questions about the end of the DNA, telomeres, telomerase? Yes.
So in that part of the language, you lose,
You lose genetic, if this is the very end, you'll lose genetic information here too. So you're losing it from both ends at the same time, every time you divide. Okay, we are. Okay, here then.
I had a question about, against long range, like, I know you just explained it like right now, but I still don't understand like the overhang and like, how does long range prevent, the loss?
Okay, cool. So this part's actually a little bit easier for me to understand, so hopefully easier for me to explain. So remember, whether you have telomerase or not, every time you divide, some of the information is going to get lost. So what the cell decides to do with this, in the cells where protecting the DNA is very, very important, it has telomerase activity. The whole idea is that you're just going to make the telomeres super long and keep making them long and rebuilding them long. So that when we lose information, it's not going to be a problem, because I can just keep making my buffer zones over and over and over again. So what the telomerase does, it attaches here and then uses its RNA template that it has within its enzymatic site. And then it just moves along in the 5' to 3' direction. So it would look like this. It keeps on moving down the 5' to 3' direction with the little RNA template. And it makes the 3' overhang. Makes the 3' overhang even longer. Not it. Yes, go ahead.
Would this be technically like telomerase having actually in place activity? Or how would you expect that to this as?
It would
Because it
be,
is adding nucleotides, right?
yeah, it's almost like I would call it DNA extension activity. It's like, it does what DNA polymerase does, except you don't need a primer. That's what it has the ability to do.
Right, the time is building.
Yes. Yeah,
So just to clarify, so that, in the first one, so that overhang is the telomere, and then what's happening is that the telomerase extends.
extends it, it makes more repeats.
Okay.
Yes,
But that part, that where the sense is not have genetic information, so it's not important, like, okay.
just random repeats. So now that I've extended this overhang, you can use your regular DNA synthesis machinery to make more DNA and fill in this gap. So you'd have primase come in with a primer, then polymerase and all the other things you need to basically extend the DNA. You get a nick, ligase comes in, fills in the nick. You get rid of the actual primer that is there. You, of course, you still end up with an overhang, but now you have so much more buffer zone from the important genetic material that's over here. Okay, so is that a little bit more clear? Okay, good.
Because some important genetic information be like copied over that has to be accounted for, or usually the buffer zone.
Usually buffer zone is pretty big, like kilobase pairs,
What?
like the telomeres go hard. Okay, any other questions?
Let's just determine from which telomeres are. Like that's.
Yeah, so what the repeat is dependent on the primer that's in that specific telomerase. Any other questions? Yes.
Kind of extension, so is that why they had the idea that once we start running out of activities,
The death, yes, that's exactly why they had the idea. Yeah, that's, 100% why they had the idea, which I mean, logically, it makes sense.
Yeah.
And then like in vitro also made sense, like the cells couldn't handle, like once they ran out of telomeres.
Right? That doesn't-
Any other questions? Okay, so really those are like the two main concepts, just the three concepts we did today. We did telomerase activity. We did the mechanism of DNA ligation. And then just basically all the stuff that you need for DNA synthesis, including looking at data for a helicase 503 prime. That's what we covered today. And that's when you ready to ask questions about all of them. If there's any other questions, let me know now, forever hold your peace. Okay, so since we hit most of the contact questions, are there any other questions about the course, the exam?
Oh, this is kind of about, they had like perfect in this lecture, I guess, but it was like for PCR, you know?
Yeah,
I like my discussion, so we like kind of went over it,
Right,
and so
cool.
we didn't go over the lecture, but, I was just wondering, like, the different conditions of PCR, like, how it would affect the actual reaction, and I was looking at the homework, and there's one where it was like, when you add like twice as much of the salt buffer
Mhm.
as you're supposed to, and, so like, from my understanding, you're supposed to add like a small amount to help DNA immune. Is,
and so the salt concentration, they usually like the magnesium salts, but then the other salt can like we talked about, if you think about the interaction between the two strands, and we were talking about salting in, salting out, it can affect the the melting temperature.
yeah, yeah.
So you basically can impact the melting temperature of the probe if you have too much salt.
Mhm.
And so, yeah, it's not specific whether you're gonna affect it rather bad or good, but definitely if you have an optimized protocol and you have twice as much, it's not gonna be something good,
You like get less of, what you want.
what you want, unless your product, PCR just didn't work as well.
And then like, it was like, that was like the first half of the question, and it's like, kind of like, in what way, like,
Mhm.
so I know that the result is that this would be less than what you want. But for how, would that mean that like the binding would be altered then, because the interactions wouldn't be like the same?
Mhm.
Like,
Okay, no problem. Yeah, so you'll notice that for homework, I count this week 3, for homework 3, the deadline's actually Wednesday night as opposed to our normal Monday night, because the questions in it cover this lecture and also some from next lecture. That we have like more time to go over it. So you can start the homework, you can look at the questions. I think you have like two full, yeah, you have 2 attempts. How many of you are doing homework by yourself or like with groups? Like what's your, by yourself? On both attempts?
Yeah.
Oh, interesting.
I prefer it.
Yeah.
well, normally I like start it and like, your questions.
Yeah, yeah.
and all this hours.
Because I think what I would do is like, I would start it, go through all the questions, and then be like, what was I sure of, not sure of, then go get feedback from like people. And then like submit, and then probably go back to a second attempt. Like that's how I would, that's why I don't normally approach stuff like that. But as long as it's working and you click grasp the concepts, like you're good. Yes, you have a
I
question.
have a question, just, in the reading guys, just, to go through, but so we went through most of the enzymes that we should know for denitrification of mitochondria already, right? And then for, the main ingredients of PCR, I was saying, like, obviously, there's like your target DNA sample, the primer pairs,
Mhm.
the DMTPs, Taq polymers, which is a special type of DNA polymerase that's heat sensitive. And this is all used to amplify the DNA, but, for the primer pairs that are used, could that be DNA or RNA for a PCR?
It can be DNA or RNA. What you'll see is that usually people use DNA.
And
Because
then,
it's more stable and you're heating and cooling, and you don't want to get hydrolysis of your primer. So for this course, and effectively for any lab that you ever go to, if you're going to design primers, they're almost always going to be DNA.
and then when it comes to, when asked like, how's a PCR reaction affected by change the percentage of like AT or GC nucleotides? So I was saying like, if there's an increase in GC nucleotides in the primer, then GC has more H bonds than AT, so require like a higher annealing temperature to bind with the DNA sample. Maybe
Yeah. So in the actual primer, the GC content is basically you want higher affinity for the primer than you have for like the DNA to the DNA. And the reason being is that you want the primer to bind and not the DNA to bind. So you're trying to have preferential binding of the primer to the DNA sequence as opposed to the DNA annealing with itself, because the complementary strand is there, right? You want the primers to lay down first, so that you extend only that portion. And we'll do PCR on Tuesday.
so more, so I guess like more GC just helps it get more stronger, like.
Interaction, exactly. Yes, question here.
So I was like, PCR, but, you said like the temperature is set high, so that it it will anneal to the primer rather than the complementary, right?
Yes, we want a higher melting temperature. So
Right.
with PCR, we have cycles of heating to break everything apart and then cooling. So as we're cooling, we want the primers to start, let's say this is primers and we got a whole bunch of these. And this is our long DNA strand and our long DNA strand. Right now it's hot. Oh, it's hot, hot, hot. I'm so sorry, stuff was in my head. And then so it's hot right now, so it's separated and you have all these primers around. If these have really high GC content, as you start cooling, you expect that these primers will associate where they're supposed to associate. Instead of these two begins associating first, because the melting temperature between the primer and the sequence, the primer and the sequence is higher than the melting temperature between the strand and its complementary strand.
So if you were to set the annealing temperature lower, then would that mean that this, if you wouldn't get the primers annealing now, you would just.
Yeah,
it would just
these
be
are
the
probably
difference
fail.
in-
You just keep like getting the actual DNA, like hybridizing with each other, and then these never get combined.
Would it ever be possible, sorry, there's like another whole thing. It's like if you set it lower, and I was thinking that, oh, you wouldn't get a product, right, because it'd just be the two.
The two DNA strands, yeah.
But I was wondering, because one of the options is also like you get multiple products, would that be possible?
Yeah, it is possible. So you can
it would
even
be
get
like
things-
some primer and some,
Yeah, you get a mix depending on what the melting temperature was. So really the whole idea is that you want a melting temperature that's higher for the primers than it is for the DNA and sufficiently high enough that you're not gonna get random hybridization here or even like a mix. So say, for example, you could have like, because like we don't know the sequence can be different depending on even the ends of the strand. So say this part associates because of the melting temperature, but then this part doesn't, then the primer associates here, then you get some replication over there. Like you see how you get different mixed products depending on where the primer is. And then because these are actually pretty long, you're amplifying different portions, you get multiple products.
so the temperature was just like some in between, like. Temperature that wasn't like ideal for,
Yeah, that's terrible,
There
terrible
is-
PCR design.
okay. Thank you.
Yes,
I know she posted the reading guides for the remaining 5 weeks.
yes.
I know like, I guess they're kind of allowed like a Monday, Wednesday, Friday schedule instead of Tuesday, Thursday. I mean, is we just
What's that?
some of the same questions or?
It's the same questions, I posted the wrong one then.
Oh, okay.
Yeah. But it's the same questions for my Tuesday, Thursday. So one thing that we do, like me and Dr. Sato and Dr. Holbrook, Inlay, Janoko, for every single section, we'll do like a group meeting. And we go over the content. We're trying to make sure every student is learning the same content that's important for them to move on to upper divs. And then after that, it's like, wow, let's free for all, and then we execute the way it makes sense for our teaching styles. But that's why you'll see like similar reading passages, similar content, similar slides, and share stuff amongst the sections. Then we all write our own examples. Okay, any other questions? So kind. So kind.
Okay, I got you. You think about that one. Fine. I have a question about, melting temperature and specifically like the, raw amount versus ratio of GC bonds to AT bonds, right? Is it more about the ratio of AT bonds to GC bonds, or is it just straight up the number of GC bonds?
It is both together.
Or is it both
Together.
tricky?
It is tricky, but it is both together.
Okay,
So like, say if you had a super long strand,
yeah,
and then you have like so many GC bonds here, GC, but the fact that it's like not dense, you still have all these areas where you're gonna have low melting temperature. But then if you have a short probe, and then the GC content is high over a short period of time, GC, then it's a really like high percent.
yeah.
And then, so even if I had like the same total content of like GC between the short probe and this long probe, this of course is gonna have way better affinity and specificity because in a small, I guess, a small space.
Right, if I was, okay.
Yeah,
The reason why I asked, well, part of the reason why I asked is my always wondered this, like I just didn't have clarity, but that's my bad. But the other reason is there was a question on the exam, where it was like, look at these DNA strands.
which one has
Based off
to,
their sequence, which
Mhm.
one would have, the highest melting temperature? And like narrowing down to 2 answers,
Mhm.
one of them technically had a higher ratio,
One of them was shorter.
A to GC, and the other one just had more GC bonds, like.
Yeah, one had more GC bonds and it was shorter,
Shorter.
and it was the length. So the length also matters.
Yeah, the length.
Yeah.
Okay.
And so the more you have to associate with each other, the higher that temperature.
It's like, how do you know though, like, when the temperature is basically bought and then the way?
Yeah,
Like,
so
where does that?
Experimentally,
Yeah.
it has to be calculated.
But on the
Like,
test-
and of course, it's obvious enough based off of the different interactions.
Okay.
So what happens is that sometimes students can overthink it, but if you're like, okay, short, lots of GC bonds, that trumps like whatever else you have, type of thing. But I mean, I think, was that the
No,
homework?
it's like-
There's a
it's like
low one?
long one.
That was the other one. Oh, yeah, I can pull it up. Okay, which question is it?
It was the one like, it was just like, all these,
I
these
was
like
all
various.
about-
Okay, oh, wait, is this exam?
Yeah.
Okay, I can't flip the exam one yet.
Oh.
So there's still one person taking the exam, he doesn't start taking it for another like 30 minutes.
Oh.
Yeah,
Yeah. Okay, I didn't know.
okay, yeah, not a problem, that's why I flipped out when people said they couldn't
But
see
can
their.
I do an approximation of it? I think I.
So I'm gonna go through every single question on the exam and say why the answer is the correct answer.
Okay.
So you'll know why, and
Yeah.
you'll have the key, and you will see your answer and the
Okay,
correct
okay,
answer. Yeah, it's just the other people who want to, like, it gives everyone the ability to take the exam with like crap happen, you know?
No,
no,
yeah, I
yes.
know what it is.
Oh, okay, cool. Now that I understand. 'Cause At first I thought it was, because I was thinking the homework question, since you're asking about it.
Yeah.
The homework question, I think, was the inverse of what's on the exam.
Oh.
Because the homework question was like, which one has the lowest TM, and I think the one at exam is highest TM. Right, cool, yes.
You told us why, we need the cameras again, you said it was because, there's no free or thing, but like, why not necessary?
Okay. So one really important thing when we talk about DNA polymerase is that it can't add a nucleotide to nothing. It just can't do it. This is a function of DNA polymerase that you have to memorize and know. And because it cannot add on a nucleotide to nothing, you have to have something there. What is cool about primase is that primase actually has the ability to start making strands from nothing. So since it can make strands from nothing, the primase creates a primer, which is RNA. It can essentially take like two nucleotides, so let's just say it's a nucleotide, and put it together and just start an RNA strand. It can just start one. Polymerase cannot do that, which is why we have to have a primase with primers to put something down that allows us to have this 3' OH that can then attack a 5' phosphate. Okay, any other questions?
See, when you have a coding DNA, you go by codons, and so like they're a trio of bases.
Yes.
So how in the non-coding DNA, how does it work? Like, how does it know? Is it also like, I'm just confused, how is the specific sequence can be read not in the codons compared to the negative?
That's actually Dr. Emily's question, because honestly, I don't know. Yeah, so once you get past like genes that are coding, like the long non-coding RNAs, even like how to sub-processes and mix sRNAs, some of the splicing stuff, like it's way out of my wheelhouse. And I'm actually, no, I'm just serious. So like my laboratory, just on the side, so I'm actually immunologist by training. And then so my lab studies most things at the protein level. So looking at protein expression, what's on the surface, what is secreted, how are they talking to each other, how are they signaling, Western blots. And we do very little molecular biology unless we're re-engineering this immune cells to say express something else, or say like CRISPRing a gene that's an immune gene into like a mouse. And so these are where we use these tools. Like for example, we're actually doing TLA analysis. One of the undergrads from 99 last year joined the lab and is using what we learned in the class to actually figure out where a transgene is inserted into mouse genome. And then so understanding how we go from from DNA to RNA to protein is very, very clear and logical. And then in this course, we don't actually touch a lot on those long non-coding RNAs and the ones that aren't coded, but there are some really cool labs on campus where like this is like their bread and butter. So there's this, let's say, yeah, he's on School of Medicine. His name is Ivan Marazzi. You can probably look into some of his research if you're interested, but like really talking about how these different RNAs like interact and are like produced and regulated. I think it's so fascinating. It's a whole different set of proteins than what you actually use for the coding DNA. It's like it's a whole other field in and of itself. So that's a cool question. Can I say anything about it? Nah. Yes.
Quick question for the exam, exam two, would you like have a question template and have us fill it out like you had a speech today for the second, give like the 5 times, 3 times.
Mhm.
We would have something like that.
Yeah.
Yeah.
Like the basic thing I want you to know how to do. So because I want to know that you can do it, I will test it. Okay, and any other questions? Any questions about the course life? How you doing? Yeah.
So, I know that like generally like people kind of like, they want always gotta be left or something, but then like, they don't really talk about like the student race, like, is that like something that they bring to doing anything like?
So the telomerase is opposite. That's the one that makes the telomeres longer, the telomerase. And so I drew it a little bit. So I want you to be aware. Once again, 3' overhang, 5', 3'. I literally have to draw this every time. Like my brain is like working so hard to keep it straight. I know it's just complimentary, but I don't know, the two numbers is crazy. Okay, so telomerase acts to essentially extend this 3' overhang on both ends of the DNA. And it has its own RNA template. Let's say this is RNA. Use the RNA template to extend the DNA for multiple repeats of whatever this template is. That's what it does. And then that's how we make the telomeres long. So telomerase is the enzyme that makes telomeres. Yes.
There's a section related about like the stem cells, like the issue of what the various connections.
Yeah.
And talk about what is the treatment and how, like certain ways to data.
Okay, so because cancer cells basically express a lot of telomerase, and usually our normal cells don't express it. As a field, we thought this could be a really great target for treating cancer. It's like, okay, if we get rid of telomerase, then we should basically reduce the longevity of these cells that are highly proliferative. They're going to lose all this DNA content, they're going to die off. So some of their goals and some of the treatments and therapies you'll see just kind of reading in the textbook that they try to target telomerase as a way to cure cancer. And vice versa, people were trying to find ways to induce telomerase expression to make you like live longer. Living longer thing didn't really pan out very much. The telomerase one seemed to have like a little bit of side effects, if I remember correctly, or so this from what's been published. And so it doesn't turn out that like targeting telomerase either by like knocking it down or overexpressing it has actually been like the great boon that we really thought it was going to be. We learned a lot about telomerase and how like cells like protective cells, but we haven't necessarily from understanding telomerase been able to design therapeutic interventions that are super productive. Yeah.
Oh, wait, so, reducing like telomerase, like activity and everything. It didn't help at all with.
I mean, not that much.
Like, it's
like
like interesting.
Yeah, it's like the outcome wasn't what we expected. So using all the information we had, the logic made sense and you test it and sometimes,
Doesn't.
I mean, well,
Or
it does, but cancer cells have multiple ways to make sure that they can live long.
Oh, there's like overload.
Yeah,
Oh, I see.
so like telomerase by itself knocked it down. It wasn't like, oh yeah, we did it, we cured cancer, it was so awesome, we changed the whole world. I mean, we were hoping it was. And every time we find a good target, something like this, where it's specific to a cell type and not made in our normal cells, it's a beautiful idea to target it because you're not going to get all these side effects from like your normal cells that don't express telomerase, right? And it just didn't pan out the way we had hoped.
What year was it?
Oh, how old was I? I'm trying to count back. It wasn't, it's maybe like 20, like it's not too long ago. I'm so bad at dates. That's one thing you'll learn about me. Like history, names, dates, there's nothing in here. But I felt so bad because like, I don't know, nothing happened. I don't know, like I made like a wedding album for my mother-in-law and I threw the wrong date on it. I'm like, it's like, oh gosh, that's bad. Yeah, I got my issues, it's fine. Yeah, actually, I think I'll look it up and see what it was. But yeah, it was really disappointing. Yes.
My, well, not, basically.
What was so really interesting, what is that covalent bond mean, that like on the ligase?
Oh, covalent bond is made between the ligase, it's like a lysine group. The lysine group makes a covalent bond with the, let's see, this is lysine. And there's like a ammonium group on it. It makes a covalent bond with the AMP when we kick off the two phosphates. So there's two sets of covalent bonds. One of them's going to be between the lysine and the AMP. Other covalent bond is going to be between the AMP and then the monophosphate that is at the 5 prime. Okay, so 2 places where you get covalent bonds. Yes. Oh, yeah. Okay, so remember we talked about pole one as like one of our example polymerases, that's the first one that we kind of discovered. And every concept that you know from polymerase one applies to all other polymerases. So pole one has 3 different enzymatic sites. I'm gonna try to draw this hand thing. Wait, this- Right? That's good, okay. 3 different enzymatic sites. We're gonna have the main one where we're extending DNA. So this is our polymerase function right in here, and the NTPs will come in through the left side. But we also have towards the front, this 5 prime, 3 prime exonuclease activity. Exonuclease. We also have down here our 3 prime, 5 prime proofreading activity. So if we're going to use this 3 from the 5 prime exonuclease activity, we can actually use it almost simultaneously to building DNA strands, because as it extends, say this is an RNA primer, you can actually eat it up and get rid of it. And so RNase H is essentially an enzyme, an enzyme in bacteria that does the same exact function. It's just not connected to a polymerase. Yes.
So in lecture, when we ask, polymerase one slash RNase H, they use one nuclease activity, and then like try to do 5 activity. Did you feel like technically, because we have to do the 5 nuclease, we can also do the 5?
No, because what will happen if it's doing the proofreading, it actually comes off the DNA strand, realigns itself, and then goes backwards, takes it off.
Ah,
You're moving the primer, you're going forwards with DNA synthesis.
and we can just like they draw the arrow from 3 to 5.
On which you have to figure out,
Maybe I'm just looking at it wrong. I'm probably just thinking it's like. This is the one that is, we extended talking about this is, this is an esoteric food. It's not actually doing anything in this paper. It's not doing, thank you.
no problem. Yes, technically not, because it starts where there's a nick. So it's at the end, it's eating from the end. So both of these are exonucleases. Any other questions?
That's probably the last example.
This background information about the 3 sites was in the prior material, and then it's recovered again for this exam. But in a little bit more depth in the process of, in the context of DNA extension and elongation.
Are we gonna go down more next week or?
This line, you know,
Okay. Thank you.
yes.
Sorry, to go back to like, again. I remember what this was about why the ends. And I know the like there's like that's why.
Mhm.
I don't know. Like why can't we just put it in there?
Well, you can put one there, but then you have to get rid of the primer. It's the loss of the primer that's the issue. Because you have to get rid of the RNA primer, it's not DNA.
Can you like draw out like where? Because I guess I don't understand that they start.
Maybe let's try to make a replication for it. Let's go. Okay. So. And depending on, so this lagging strand. Like you should be able to keep on basically filling it in all the way up into the end. Because you can see that the primers are back here, they'll get deleted. And you'll fill it in 503 times, it shouldn't be an issue. Then on this leading strand, if this is the very edge of the DNA. and you had a primer here because you needed a primer to start, then as you replicate, you lose information because you have to get rid of the primer. And you can't fill it in because there's nowhere to start the DNA replication from.
Oh,
So depending on where like this primer lands down, so like if your primer starts exactly from the end, then yay, we didn't lose any information. If your primer started like right here, then you're gonna lose whatever information is back here.
it's fast. It's not like the ends are. Sometimes it's not really hard to end.
Yeah, the primers I make are exactly the end, like where we want it to point. So we control what gets amplified. And we're actually gonna go through the steps of PCR on Tuesday. Yes,
Oh, yeah, like, it's all happening, the process of that,
Replazone, yeah.
so, like, is it happening in the body, like? do they know-
So this we're actually talking about like eukaryotic. This was kind of happened in a bubble too, like a little replication bubble, but what we're talking about is some of the eukaryotic machinery. Or actually, no, we do prokaryotic machinery and it gets more complicated in eukaryote. And then so this actually does happen in a replication bubble. And then it just kind of keeps going around. And so we talk about, when we say bubble, we mean bacteria. When we say fork, we mean eukaryotes. And then for principles, we're talking about what it looks like at a replication fork. And because we also want to talk about telomeres, telomerase, so what's important for the ends, we're talking about eukaryotic. But for principles of the basic mechanism, you see that we jump back, especially to like a prokaryotic protein where it's very straightforward to understand the basics. Yes.
There's this one picture in the board, if you look at the one twenty four, like, picture, one of the transfer, how I was doing, and maybe trying to move, people and whatever. It looks like, this is like, real pretty good, so, I didn't understand it, but at all, with, what the band name and the back.
Oh, okay. So if
So do not.
we don't need to know it, I mean, it's actually, it's a really good experiment. Right,
Might as well.
it's like, sure, yeah.
But it's a lot of fun.
Yeah, it's actually, it's very complicated, but it's so elegant. It's- You know what? I'm gonna take that, take that new discussion.
Yeah.
Yeah, I'm gonna put it in the discussion, because like now, for the elegance, like it so beautifully demonstrates like how it works. And I think if you can understand the figure, but this thing, like I don't test on knowing about the clamp and having to come on and off. And like that's what that figure basically explains, is how do they find out that, okay, the baby chunk is like keeping it on there. And that's what that figure does. And you just need to know that the beta clamp keeps it on there. It's a very straightforward like thing to know without having to go through the experiment.
No.
Yeah, I was gonna, okay.
That is so cool.
It is cool. Okay, I'm gonna put on a discussion worksheet. But no, when it goes on the worksheet, it becomes fair game for exams. Which is. We'll see how I feel. But I think I'll still put on the worksheet. It's very beautiful.
Week 3 or
It'll be for week 3. So you'll see it, discussion starting next Wednesday. Yes.
From how he was straight on the game and I protein or which I can do like after it has like how it's gonna be after both things so combines it, how does it SSP with the work?
Yeah, so the mechanism is actually super easy. So once you have DNA associated with protein, it's less accessible to nucleases. So if nucleus can't reach it or access like sites to cut it, it doesn't get cut. It's almost that same idea as when it's wrapped around the histone. Remember how the histone protein protects it? So when it's single stranded, we're like, oh, we need to protect it, you associate it with a protein. Yes.
Yeah, on that, when we're talking about assessments,
Mhm.
the book mentions something about all the results.
Oh, yeah. Okay, so you'll talk about hairpins with Dr. Inlay, I believe, I know some of his material. But a hairpin is like a secondary structure of actual RNA. So sometimes you can have RNA or DNA, and when it comes single stranded, depending on the sequences, you can form what we call these hairpin structures. So parts of the DNA that aren't like the complementary strand have enough of a sequence that match each other that will stick together if all these weird loops. But the association of single-stranded binding protein is so strong that it'll open these up and wrap around associated with the protein and break these interactions. Yeah, it's also not in the exam, but it's a great question. Yes,
So, there's like, just the end loss situation, you know what I'm talking about.
an end loss situation, y'all. Let me start calling that end replication problem.
Thank you. The end replication problem happens primarily at the terminal lagging strand. It doesn't happen like on the leading strand, right? Because the leading strand starts in the middle of the replication fork, so there's gonna be, you know, that other.
It only happens at terminal strand period. Yeah. And so sometimes you start replicating in the middle and the fork can be in the middle, but remember like that little nick, like ligase will come behind and fill it in. It's the terminal end of the actual DNA is where you're gonna have the end replication problem.
Gotcha.
It's not about where the end of the fork is, it's where the end of the DNA
Oh,
strand
that's
is.
true. Okay, thank
Yeah,
you.
No problem.
Gotcha.
Okay, you had a hand back here? Change your mind?
Oh, I was trying to get the protein for it stops the like supercoiling into working DNA, but it's the single stranded binding protein. It's like.
Yeah.
Okay, it's the same thing. It prevents any,
Oh, you mean the topoisomerase?
is that what stops
Okay.
the coiling?
So when you're taking helicase, yeah, so just think of, okay,
That's supercoiling.
you, you know, when you have, like the olden day telephone cords
Yeah.
and like twisty. And if you try to like pull twist out of it by straightening it out, at the back end, it's super tight and twisted. Yeah, so topoisomerase, what it does, it goes snip, boom, release the tension. That's what topoisomerase does, releases the tension in that coil because of the unwinding by the helicase.
So the supercoiling releases the tension in the single stranded binding protein. That's what it's single stranded.
Yes, prevents it from annealing and keeps it getting cut up by nucleus. That's a good question. Yeah, we didn't topoisomerase in lecture. But it is an important part of the process. Yes,
How does the term location work? Like, I'm just confused about when it combines onto one strand, like, how is it transitioning on the
so it causes, so it's actually like a protein with like 6 subunits, and you get conformational changes that causes it to rotate with some torque. And that allows it to like basically spin, and that spinning force is what's pushing the DNA apart. Like I said, I don't really like the diagram that, I'm not gonna find a better diagram for this. Diagram that we have about the function, because in the diagram, it looks like it's doing like that's how people like to imagine it, but that's not really how it's working. So just think of conformational changes are allowing it to move along the DNA strand by causing tension and allowing it to spin. Okay, any other questions? We got about 9 minutes left. Hit the road, Jack, and I'm not gonna come back, but I will. I'll be back. All right. Yes,
In order to like answer this question, why is it, to understand my study, can you explain the 796 and the 722 one more time?
yes. Because you need to know it, and you will see questions on it. I will have to explain it one more again. One more again. Okay, so this reaction actually occurs in 2 separate tubes, where you have DNA here, this DNA is not labeled with any. You add in helicase, you add in your ATP, which will allow the helicase to do the function it needs to do. You add a probe in one tube, a probe in another tube. These 2 are labeled, they are 2 different lengths. They are radio labeled so that when we put them on a gel, we can see them. After we allow the actual experiment to go to completion or to run for a little bit, we run them on an agarose gel in 2 different lanes. In lane 1, we're going to run this reaction. In lane 2, we'll run the other reaction. When we run this first reaction, we expect to see that there's some of the reaction that didn't go to completion or just didn't happen. So we always expect a high band for the most part, meaning that we're seeing this big piece of DNA that has a probe on it. But then if the helicase is moving in some direction, whether that's 5' to 3', or 3' to 5', it's gonna knock off the probe at the end, depending on which way it's moving. If it's going 5' to 3', you'll knock off the 722 tumor, because it is small, you'll see it towards the bottom of the gel, so you get some signal here. That tells us we have a 5' to 3' helicase. No, there's different types of helicases. This experiment says that this helicase moves in this direction. So the idea is to understand how the experiment works, So you can test what direction the helicase moves. Okay. Okay, so I'll see y'all Tuesday. Like I said, your questions for the guys will be posted tonight, so you can see what you got wrong, what you got right. If any of you were in here to do paper copies, I'm gonna go update the paper copy grades and then just email me for coming and look at your test. No problem.
Oh, for this one.